Hello and welcome to this presentation which marks 10 years of research in ME/CFS. I am a patient and independent researcher and I have been using various computational methods to research ME/CFS. The presentation is aimed at patient organizations, researchers, but also patients themselves. Now, before continuing, some disclaimers. And here is my background. I am a data scientist and a machine learning consultant. I am a patient now in remission. I have 2 publications in ME/CFS and long COVID research, and I'm also a patent owner of AI-assisted medical research methodology. So here is our agenda. Present findings from 10 years research work for ME/CFS. See which areas emerge as research hotspots given latest findings. And finally, put everything together and propose a hypothesis. Here are the research targets that I have been looking at. This is the first page, and here is the second page. And obviously, there's an urgent need to look at the bigger picture and to try to understand what do studies show us and whether they converge on a hypothesis. The other thing is that we must look at is whether we have a single point of failure or not. So the big question, do we have convergence? Here are the key factors, the key topics that we need to be aware of. Metabolic bottlenecks and metabolic disruptors, lipid metabolism, cell membrane integrity, calcium and cholesterol metabolism, mitochondrial vulnerability, vesicle trafficking, ER stress, this is endoplasmic reticulum stress, proteostasis, glycosylation defects and especially N-linked glycosylation, immune antiviral regulation, oxidative stress, and finally glutamate metabolism and brain synapses. So let's look at the metabolic bottlenecks. We see the first part from a file which I will provide a link for you to download later on. Under the column name, we see certain conditions and we also see specific treatment and diagnostic markers for each condition. So the question here is how many of the concepts being presented in this file exist in ME/CFS. So we start with nitrogen. We see an entry of disorders of nitrogen containing compounds. Dr. Chris Armstrong is looking at the nitrogen hypothesis. Here is the part 2, and in part 2 of the file, we see Wilson disease, an entry of Wilson disease, and another entry of hereditary hemochromatosis. Now, on the left snapshot of this slide, we see a part from the GWAS Decode Me candidate genes, and it mentions hereditary hemochromatosis. On the right side, we see a study mentioning Wilson disease and its relevance with long COVID. Here is part 3. We see several entries of glycogen storage disease. We also see an entry for hereditary fructose intolerance. This is part 4, and we see 2 entries of POG1 and POG2 deficiency. So we see a case from a fibromyalgia patient, and this patient was found to have several POG1 polymorphisms. This is Paolo Macalini. This is a page from Paolo Macalini, GitHub, and in an analysis that he did, he found ATP8B1 to be a gene of interest. Now, if we look at part 5, we will see ATP8B1 deficiency. Another entry shows disorders of peroxisomes. And we know that in ME/CFS we have peroxisomal dysfunction. Another study related to carnitine palmitoyl transferase 1 and 2 deficiency, and it has to do with chronic fatigue syndrome. In part 6, we see 2 entries of this specific condition and also observe that we have specific treatments. So in this case, we need to do frequent feeds, have a low-fat diet, and use medium-chain triglycerides. Another page from ME Association, this time June 2020. It mentions hemin and porphyria. And on the right-hand side of the slide, you can see a study suggesting a protective effect of hemin against experimental chronic fatigue syndrome in mice. So let's see whether porphyria and hemin exists in this file, and it actually does. So this file exists, and you can see the link at the bottom of the slide, and it's about metabolic liver disorders. And the question is whether we have been looking at several signals suggesting that the liver is involved. Here is a study by Germain et al., and they have found a reduction in compounds which are suggestive of damage to the liver. A newer study by Bingius et al., and they say that they find a combination of chronic inflammation, insulin resistance, and liver disease. So let's move on now to lipid metabolism. Here we see 2 studies showing phospholipid and peroxisomal dysregulation. In this point, let me comment on the fact that in titles before and here we see red stars among the titles. I will explain why these stars exist. Another study suggests dysregulation of bile acids, this again from Maureen Hansen group. This is from Tamara Karnak, an independent researcher, and she identifies in a hypothesis that phosphatidylcholine deficiency might be important because it disrupts cell membrane integrity. So this is a study and it relates to total parenteral nutrition. So this basically says that every patient that is under total parenteral nutrition should be supplemented with choline. So here is a study, again, Maureen Hansen, and we see that among other compounds, we see issues with acylcarnitines. So let's keep that for the next slide. Again, this might all be a coincidence, but we see that this is a page from the American Association for the Study of Liver Diseases. And as I mentioned, we see also acylcarnitin and several other concepts, for example, lactate, pyruvate, amino acids, urea cycle, and all of these are liver related, and we are finding them in ME/CFS research. So I will have to show a slide for lipids, and because I think it's very, very critical. Lipid absorption is very critical. And so lipids are crucial for cell membrane integrity, and especially phospholipids. Lipids are key building blocks of cells, including immune cells. They regulate immune initiation and resolution, and they require, mitochondrial membranes require lipids to function. Cell membrane integrity, something I believe very crucial, so I confirm from my research what Tamara Karnak suggests. Intact membranes regulate entry of nutrients, glucose, amino acids, exit of wastes, and movement of ions and water through channels, pumps, and transporters. They keep distinct ion concentrations, sodium, potassium, calcium chloride, and these gradients power nerve impulses, muscle contraction, including heart rhythm, and many transport processes. They help maintain cell morphology by providing structural support. And here a question is whether red blood cell deformability actually originates from cell membrane integrity disruption. Finally, we see that the glycoproteins are mentioned. And they are mentioned because they might be very important in ME/CFS. This is a network analysis that was generated in 2017, and we see different concepts being associated with each other. On the left-hand side, we see cluster 1, and cluster 1 has concepts that are related to metabolism and oxidation. On the right-hand side, we see cluster 2, and cluster 2 has concepts related to immune system and inflammation. And in the center of these clusters, we see glycoproteins. Now, glycoproteins are actually proteins that went through glycosylation. So let's keep that. So are glycoproteins a critical hub? They sit between metabolism, immune system, oxidative stress, and inflammation. They influence cell signaling, trafficking, receptor sensitivity, and immune tone. Finally, the endoplasmic reticulum and glycoproteins are directly related. We move on now to endoplasmic reticulum stress and end-linked glycosylation. Again, I will have to remind you that glycoproteins are glycosylated proteins. So we see the first study mentioning ER stress in 2023, a newer study by Maureen Hanson finding endoplasmic reticulum stress in post-exercise males with myalgic encephalomyelitis, chronic fatigue syndrome. And from this study, we find a mention on disulfide bonds and their importance for correct protein folding. A study again by Maureen Hanson group, and in the study we find the mention of N-glycans and how these are necessary for glycosylation and protein folding quality control. Here's a study which I'm really honored to be a part of, and I would like to thank Dr. Wenjong Xiao and Kimberly Hicks from the Open Medicine Foundation for having me aboard. And in this study, it was found that there were low asparagine levels, and asparagine is key for proper end-linked glycosylation. So if we look at part 5 of the file that I mentioned in the beginning of the presentation, it's very interesting to see that under congenital disorders of glycosylation, we find disorders of end-linked glycosylation. Now, of course, I do not say here that ME/CFS patients have something which has to do with congenital disorder of glycosylation. What I propose here is that patients, ME/CFS and or long COVID patients, and some patients of other syndromes as well, potentially, may have issues with impaired end-linked glycosylation, but they're not so severe to show off very early on in their lives. So, ascorbyl palmitate, I would like to thank Violetta Q. Smith, who showed this tweet by David Feigenbaum, and Dr. Feigenbaum has used ascorbyl palmitate to treat children with congenital disorders of glycosylation. Now, why do I believe that this is important for ME/CFS? Because ascorbyl palmitate provides antioxidant support at the cell membrane. And so anything that looks and improves oxidative stress at the cell membrane, I think it's very important. So some glycosylation cofactors, we need to be aware of these: zinc, magnesium, calcium, manganese, and dolicol. And this is done by polyprenol supplementation. So because of all of these mentions of glycoproteins, end-linked glycosylation, I think that we should be looking forward to results of this study exploring the glycome, and this study has been funded by the We and ME Foundation. Proteostasis. So we have a deep learning analysis by Zhang et al., also Fereste Jahanbani in the study, in Michael Snyder. And they mentioned the ubiquitin proteasome system protein degradation in this study. And I think that we should be looking again the connection of the ubiquitin proteasome system with endoplasmic reticulum stress. This is from the mecfsscience.org blog. And in this post, we find many genes that originate from Decode Me GWAS study, and some of them have to do with glutamatergic signaling. This is a study about ubiquitin proteasome system, glutamate, and ATP, and I believe that one study that should be done is something that connects these concepts with ME/CFS patients. Now we'll look at cholesterol and calcium metabolism. So this is a study by Open Medicine Foundation and Dr. Chris Armstrong. We see NR1H3. This is LXR type alpha, the liver X receptor type alpha. We see another identification of LXR, this time from Maureen Hanson group. We see impaired LXR RXR activation post-exercise in ME/CFS patients. Here I show the study by Precision Life 2025. They identified 259 candidate core genes, and this is a list of these genes. And also we see a UGGT1 gene. So definitely we need further exploratory work to do in these type of analyses. And so let's see that we have cholesterol metabolism genes in this study by Precision Life. We see ABCC6. You also observe the red stars. We see calcium channels and UGGT1 again, which is critical for N-linked glycosylation. Now, regarding calcium channels, I believe that we must investigate the role of the cell membrane. So in basically how cell membrane disruption disrupts these calcium channels, if such thing is taking place, and to also see the proteasome function and to explore the role of heat shock proteins. So trying to put everything together here, we see LXR, RXR, we see APCA1, we see MERTK, this is vitamin K related. We see here PS, this is phosphatidylserine. TSP1 is thrombospondin 1, and this is being looked at by researchers at the Open Medicine Foundation. So I would say that this chart has a lot of the concepts that we have been finding. Also, see that we have lysophosphatidylcholine, which is a phospholipid. So a study in 2021 is actually mentioning MERTK for efferocytosis and also phosphatidylserine. And basically, these are concepts that we've seen in the previous slide. So the red stars actually signify that all of these concepts have been identified during my work. So the APCA1, LXR, CYP7B1, ABCC6, and all of the rest of the concepts have been identified using machine learning and network analysis methods. So this is a circulated document in 2017. You can see very clearly that I have in the document the figure that I've shown previously. You see mentions on end-linked glycosylation, mentions on LXR, oxysterols, misfolded proteins, the unfolded protein response, and also a network analysis that was done in 2017. Again, you see here choline deficiency, peroxisomes for peroxisomal dysfunction, LXR, CYP7B1, this is for precision life, and of course liver-related concepts. In Euromen 2018, just want to take the opportunity to thank Professor Modra Murovska for inviting me in London School of Hygiene and Tropical Medicine in 2018 to present my work. And there you see many mentions on the liver function, hemochromatosis, and also oxidative stress and endoplasmic reticulum stress. This is from the same presentation, which of course was done in front of CFS researchers. And you can see CYP7B1, CH25H, and EBI2, which is related to Epstein-Barr virus. This is a tweet that I made in 2020 regarding ABCC6. I propose that this is important because it transfers glutathione conjugates. So what does it all mean? In order to try to understand what is going on, I've been looking at several computational methods and also lately reasoning engines, artificial intelligence. And so in order to make an analysis, I used the DecodeMe GWAS study genes and placed extra weight, obviously, to these genes. Then I looked also at hypotheses. And basically, this is the GWAS study, and these are the results, and these are the candidate genes. And so basically, we have a committee of 6 AI reasoning engines looking at the same problem and trying to identify and suggest a causal hypothesis. So where they all agree. They agree that mitochondrial stress and endoplasmic reticulum and trafficking stress takes place, that the membrane lipid and phospholipid depletion is central, and also that there is subgrouping involved. So where AI disagrees? Well, there is disagreement whether there's the mitochondria primarily that is the originating problem or the ER trafficking problems. And these are identified by the genes identified on the GWAS study. So there is some reality check. All of this information I put for researchers to look at, and obviously to look at every information that is in this presentation, because I'm not a medical professional. And so this is a consolidated hypothesis for patient organizations and for MECFS and long COVID researchers to look at. Finally, this is a slide to look at raw Accutane. Maybe this medication might be doing something in ME/CFS patients. Again, this is a hypothesis. I'm not making any claims or suggestions. I would just like for ME/CFS researchers to look at this hypothesis. This is an email that I sent in December 2015, so 10 years ago. And in this email, which was circulated to a number of researchers, you see certain and specific concepts that we described, and most importantly, that we have been finding in the last decade. So endoplasmic reticulum stress, bile acid metabolism dysregulation, and also unfolded protein response. So here are my closing thoughts. It appears that we have convergence between previous research and current findings. I'm really happy to see that this is taking place. Several of these topics were communicated years ago, but unfortunately, no follow-up has been made, unfortunately for patients. ER stress, end-linked glycosylation, proteostasis, glutamate dysregulation, cell membrane integrity. So these have to be more closely looked at. And finally, I look forward to working with patient organizations to advance research using computational methods. Thank you very much for your attention and for your time.